Okay, so let's continue last class. So we were discussing spermatogenesis, and in spermatogenesis we were discussing the whole story start at the age of puberty. In puberty, supraoptic nuclei and paraventricular nuclei of hypothalamus secrete hormone GnRH, and this gonadotropin releasing hormone GnRH hormone click on the process of sexual maturity, spermatogenesis, or in female it is their appearance of secondary sexual character, means all kind of sex related things will click on by GnRH hormone at the age of puberty. Right now, this GnRH hormone act on anterior pituitary and it causes secretion of these two hormones, follicle stimulating hormone and luteinizing hormone. This follicle stimulating hormone and luteinizing hormone, also called gonadotropin, this luteinizing hormone receptors present on Leydig cell. It will bind with the Leydig cell, and then it's going to order the Leydig cell that you synthesize testosterone hormone from cholesterol. So in Leydig cell, by the help of cholesterol, stepwise, not in one step, stepwise, finally testosterone hormone synthesized. And in this testosterone hormone, it's a steroid in nature. Okay, so it will fuse within seminiferous tubule. In seminiferous tubule is going to promote the process of spermatogenesis. Okay, follicle stimulating hormone bind with Sertoli cell, and then they secrete hormone androgen binding protein. This androgen binding protein will bind with testosterone hormone, and then it's going to concentrate this testosterone hormone in seminiferous tubule. Okay, right? And now this testosterone hormone promote the process of spermatogenesis. How? Let's see. Actually, this spermatogonia, first it will increase this number, and by mitosis it will divide and increase this number, and this divided cells are called primary spermatocyte. Now this primary spermatocyte will break this tight junction, right? They will break this tight junction, and then they will come out. Okay, and once they come out into the testicular lumen, this primary spermatocyte is going to divide into one primary spermatocyte actually is going to divide into two cells, and that cells are called secondary spermatocyte. Okay, and now this each secondary spermatocyte will actually go through meiosis second, and then they will form 1, 2, 3, 4. Total 4 spermatid. And now this spermatid will differentiate, and finally they form total 4 spermatozoa, right? So one primary spermatocyte go through meiosis first and meiosis second, and finally they form total 4 spermatozoa. This all growing stepwise gamete cell, either it is spermatogonia, or it is primary spermatocyte, or it is secondary spermatocyte, or it is spermatid, or it is spermatozoa. This all gamete cell is going to obtain nutrition from the Sertoli cell. That's why we are calling Sertoli cell nurse cell. Yeah. So we are calling Sertoli cell nurse cell. Clear, student? And finally, the spermatozoa, they releases from the Sertoli cell in testicular lumen, and then slowly, whenever required, they will come out of the testis. Right? The process in which this spermatozoa releases out from the Sertoli cell is called spermiation. Okay, let's talk more about hormonal regulation here. Actually, when this follicle stimulate, for anything, too much is not good actually. So even too much testosterone also is not good actually. So high level of testosterone will send a negative feedback signal to pituitary gland, and then luteinizing hormone level will decrease. Okay, high level of they are going to act here, right? And luteinizing hormone secretion will increase. Same thing about Sertoli cell also. Whenever spermatogenesis will be too high, this Sertoli cell secrete one hormone that is called inhibin. This inhibin show negative feedback, it will send signal to pituitary gland, don't secrete follicle stimulating hormone. So follicle stimulating hormone synthesis secretion will decrease, process of spermatogenesis will decrease. So this is what hormonal regulation of spermatogenesis. Thank you so much. Remaining we'll see in next class. Bye.